preserved to the nucleus from bacteria symbionts during the evolution of the modern-day organelle. 3. Mitochondria and chloroplasts contain their own ribosomes. Ribosome cell structures involved in protein synthesis exist in either a large form, the Sedberg units, typical of the cytoplasm of eukaryotic cells, or in smaller form 70S, unique to prokaryotes. Mitochondria and chloroplasts contain ribosomes, and they are 70S in size, the same those of the prokaryotes. Antibiotic specificity. Several antibiotics, streptomycin is one example, kill or inhibit bacteria by specificity, interfering with 70S ribosomal function. These same antibiotics also inhibit protein synthesis in mitochondria and chloroplast. Molecular phylogenetic studies using comparative ribosomal RNA sequencing methods have shown convincingly that the chloroplast and mitochondrion are closely related to bacteria. These studies clearly point to the modern eukaryotic cell as having arisen from an association of two organisms, presumably by endosymbiosis. Evidence that hydrogenism or endosymbionts has been found as well in obliquely anaerobic ciliated protozoa. Nyctothyrus ovalis that lives in the hindgut of termites. Hydrogenosomes have been identified that contain DNA and ribosomes. Moreover, the nucleus of hydrogenism containing eukaryotes contain genes encoding specific proteins known to be of bacterial origin. Thus, hydrogenisms probably originated by the endosymbiosis of the pyruvate fermentating bacteria. Such as Clostridium species with the anaerobic microbial eukaryote, presumably as a way to extract a bit more energy out of a strictly fermentative lifestyle. If this scenario is true, the mitochondria and the hydrogenism can be viewed as functionally related organelles that simply show different metabolic patterns. In summary, many forms of evidence point to our organelles as having arisen from the endosymbiotic uptake of free-living bacteria by eukaryotic host cells. In this cozy arrangement, host cells obtain partners specializing in energy generation, while the symbionts received a stable and supportive growth environment. That endosymbiosis was an evolutionary success is attested to by the presence, with rare exception, of energy-producing organelles such as mitochondria or chloroplast within eukaryotic cells today. A variety of other cytoplasmic structures are typically in eukaryotic cells, including the endoplasmic reticulum. Other organelles in eukaryotic cell structures: Golgi apparatus, liposomes, peroxisomes, in contrast to mitochondria and chloroplast. However, these structures lack DNA and ribosomes and are not of endosymbiotic origin. Endoplasmic reticulum and Golgi apparatus. The endoplasmic reticulum is a network of membranes continuous with the nuclear membrane. Two types of endoplasmic reticulum, ER and recognized rough, which contains attached ribosomes, and smooth, which does not. Smooth ER participates in the synthesis of lipids and in some aspects of carbohydrate metabolism. Rough ER, through the activity of its ribosomes, is a major producer of glycoproteins and also produces new membrane material that is transported throughout the cell to enlarge the various cell membrane systems. The Golgi apparatus consists of a stack of membranes that function in concert with the ER, and the Golgi complex products of the ER are chemically modified and sorted into those destined to be secreted, for example hormones or digestive enzymes, those that function in other membrane structures in the cell. Lysosomes and peroxisomes. Lysosomes are membranes and closed sacs that contain various digestive enzymes that the cell uses to digest macromolecules such as proteins, fats, and polysaccharides. The internal pH of the lysosome is about 5 to 2 units lower than that of the cytoplasm. Then the hydrolytic enzyme within the lysosome function optimally at this pH because the hydrolytic enzymes are nonspecific in their action and could potentially destroy key cellular macromolecules. If not contained, the lysosome is a structure that allows lytic activities to be partitioned away from cytoplasmic proper, following hydrolysis of macromolecules and lysosome. The resulting monomers pass from the lysosome into the cytoplasmic as nutrients for the cell. The peroxism is a membrane enclosed structure whose function is to produce hydrogen peroxide, H2O2, from the reduction of O2 by various hydrogen donors, including alcohols and long-chain fatty acids. The H2O2 produced in the peroxisome is degraded to H2O and O2 by the enzyme catalase. Peroxisomes play other roles as well, such as synthesizing bile salts that aid in the absorption and digestion of fats. Peroxisomes originate in the cell by incorporating their proteins and lipids from the cytoplasm, eventually becoming a membrane-enclosed entity that can enlarge and divide in synchrony with the cell. Microfilaments and microtubules. A large size of eukaryotic cell and their ability to move requires structural reinforcement. This support comes from proteins that arrange themselves into filamentous structure called microfilaments and microtubules. Microfilaments are about 8 nanometers in diameter and are polymers composed of subunits of the protein actin. These fibers form scaffolds throughout the cell, defining and maintaining the shape of the cell. Microtubules are larger filaments about 25 nanometers in diameter and composed of the protein tubulin. Microtubules assist microfilaments in maintaining cell structure but also play an important role in cell motility, both in the movement of internal cell structures, for example in the separation of chromosomes during cell division, in movement of the organism itself, for example in movement of the flagellum and flagellated eukaryotic cells. Together, this system of microfilament and microtubule fibers is called the cell cytoskeleton. We saw in an earlier chapter that prokaryotes have homologs of actin and tubulin in the form of the proteins MREB and FTSZ, respectively. Thus, the eukaryotic cytoskeleton has deeply evolutionary roots. Overview of eukaryotic genetics. Here we briefly consider some of the idiosyncrasies of genetics in eukaryotes using the baker's yeast Saccharomyces cerevisiae as our model organism. Eukaryotic cells undergo sexual reproduction with male and female, each donating gametes to form the zygote. This is fundamentally different from the unidirectional mating process in prokaryotes, and so we focus on these events in our discussion. From a genetic standpoint, eukaryotic cell can exist in two forms, haploid and diploid. Diploid cells have 2 of each chromosome, whereas haploid cells have but 1. Cells of S. cerevisiae can exist indefinitely in the haploid vegetative stage, containing 16 chromosomes, but occasionally 2 haploid yeast cells can fuse mate to yield a diploid cell, 32 chromosomes. This process should not be confused with the state of affairs in cells. Metazoans, such as the plants and animals, in these organisms, the diploid phase and presence in the organism proper and the haploid phase occurs only in the gametes, Recall that mitosis is the process following DNA replication, in a cell in which chromosomes condense, divide, and are separated into two sets, one for each daughter cell. In haploid cerevisial cells, mitosis occurs before each cell division to maintain the 16 chromosomes per cell. Meiosis is the process by which the conversion from the diploid to haploid stage occurs. Meiosis involves two divisions. In the first meiotic division, homologous chromosomes are segregated into the separate cells, changing the genetic state from diploid to haploid. The second meiotic division is essentially the same as mitosis, as the two haploid cells divide to form a total of 4 haploid gametes. Ascospores, figure 14.8. The mating process and yeast mating types. Yeasts have two different forms of haploid cells called mating types. These can be considered analogous to male and female gametes. On mating of the opposite type, a diploid cell is formed from a single diploid cell. A structure containing 4 gametes is formed, 2 of each mating type. The cell in which the gametes are formed is called an ascus, and the cells within the ascus are called ascospores. The 2 mating types of Saccharomyces cerevisiae are designated X and A. Cells of type A mate only with cells of type X, and whether a cell is A or X is determined genetically. Some haploid strains of S. cerevisiae remain or X, while other strains are able to switch their mating type from one to the other. This switch in mating type is caused by an inserted gene, and the phenomenon is illustrated. There is a single location on one of the S. cerevisiae chromosomes called the MAT for mating type, locus at which either gene A or gene X can be inserted. At this locus, the MAT promoter controls transcription of the which-over gene is present. If gene A is at the locus, then the cell is mating type A, whereas if gene X is at the locus, the cell is mating type X. Elsewhere in the yeast genome are copies of genes A and X that are not expressed. These silent copies are the source of the inserted gene. When the switch occurs, the appropriate gene A or X is copied from its silent site and inserted into the MAT location, replacing the gene already present. The old mating type gene is excised and discarded, and the new gene is inserted. This process has been called the cassette mechanism because the A and X gene can be considered analogous to tape cassettes. Which genes is inserted in the MAT locus is the one that will be transcribed. The X and A genes of S. cerevisiae are regulatory genes, among other things. They regulate the production of the peptide hormone X factor or A factor, which are excreted by yeast cells undergoing mating. These hormones bind to cells